Okay, good morning. So yesterday we have seen about the concept of active principle and major metabolic pathway of carbohydrate. And today we are observing the major metabolic pathway of proteins leading to the production of therapeutically active chemical constituents. So therapy means what? The act of use of the medicine, right? So in yesterday's lecture we have seen about how carbohydrates are generally showing the effect on the human body. What are the characteristics? So now in proteins, the macronutrients, as we are knowing, the organic compounds that yield the energy, and they are classified as carbohydrates, fats, lipids, and proteins. Generally, proteins are used for building and no capacity for the storage, but the carbohydrates has and the fats has the limited capacity, or fats having unlimited and they having the capacity for the storage. In these major pathways, we have seen these 8 pathways: glycolysis, gluconeogenesis, glycogen metabolism, fatty acid metabolism, citric acid cycle, oxidative phosphorylation, amino acid metabolism, and only the liver can carry out all these reactions, the major pathway. So here the key junction points in relation to this. So there are junction points are glucose G phosphate, then pyruvate, and acetyl CoA. See here how the glucose is related to this. So glucose 1 phosphate, then fructose G phosphate, and say G phosphogluconate. So they are produced or vice versa from the glucose G phosphate. The glucose 1 phosphate gives glycogen, fructose G phosphate gives pyruvate, and the G phosphate gluconate gives the ribose 5 phosphate. So this is a one normal link is there. In the position of metabolic pathway, there is having the position of in the cytosol. So glycolysis, pentose phosphate pathway, and the fatty acid pathway, where the mitochondrial matrix, it shows the citric acid cycle, oxidative phosphorylation, beta oxidation of fatty acids, ketone body formation. In the second, there is a interplay of the both these components in the gluconeogenesis and the urea synthesis. In the metabolic fat of glucose is there. So glucose, as it is the intracellular form of glucose is glucose G phosphate, and only liver cells have the enzyme glucose 6 phosphatase that cephalophosphorylates G6P, and the releases glucose into the blood for the use by other tissues. G6P can be oxidized for the energy in the form of a ATP and the NADH. Then G6P can be converted to acetyl CoA and then the fat. The excess of G6 is stored away as a glycogen, and it can be shunted into the pentose phosphatase pathway to generate the NADPH and this ribose 5 phosphate. So remember this metabolic fate of the glucose. Then metabolic fate of the fatty acids are there. These fatty acids are oxidized to acetyl CoA for the energy production in the form of the NADPH. Fatty acids can be converted to ketone bodies, and the ketone bodies can be used as a fuel as the extra therapeutic extra hepatic tissues. These fatty acids are used for the biosynthesis of the bioactive molecules, and such as arachidonic acid and the eicosanoids, which are derived from the fatty acids. The excess fatty acids are stored away as the triglycerides in the adipose tissue. Adipose tissue is the part of the nervous tissue. Then central themes for metabolic pathway, as it is discussed. The acetyl CoA is a common intermediate of all metabolic pathways, and it is interconnect glucose, fatty acids, and amino acids metabolism. This oxidation of dietary fuel, it leads to the capture of energy in the form of ATP and the NADH. So the role is the important, and the biosynthetic and the degradative pathway are the disjunct and coordinately regulated. So there is a proper regulation. Nextly, endocrine regulation of the metabolism. In this metabolic process, they are tightly regulated by the uh, concern concerted action of the hormones, and these hormones regulate the enzyme primarily by the covalent phosphorylation. The three main endocrine signals that is the metabolism are epinephrine, then glucagon, and there is the epinephrine is responsible for the metabolization of this glucose. Here the glucogenesis and inhibition of the enzyme activities of the fuel storage pathway. Then second pathway we are knowing glycolysis. So glycolysis oxidizes one molecule of glucose to two molecules by pyruvate. NAD plus is generated under the anaerobic condition. The pyruvate is reduced to the lactate by the regeneration of the NAD plus. Phosphofructokinase is the key regulatory enzyme, and it catalyzes the committed step of the glycolysis, the conversion of F6P to F16BP. It is activated by 2 F26BP and inhibited by the citrate. So this is for the glycogenesis. Then the brain accounts for 20% of the energy requirement of body, and mostly to maintain the membrane potential required to transition of the nerve impulses. Normally glucose is the sole preferred fuel of the brain, and the brain's consumption of glucose and the oxygen are important. The metabolic profiles of the brain. The brain cannot store glycogen, and it must receive a constant supply of glucose through the blood. The brain cells have the glucose transporter, then GLUT3 with a low kilometer for the glucose. In the event of the starvation or disease, the brain cells adapt quickly and utilize the ketone bodies, and that are made up in the liver. So if anything is this, the first attack is to the liver is there. So this is the one part. And second is skeletal muscles are there. So these muscles are different types. So first type, suppose the we just see, or the development on the other hand, the muscle can store glycogen. Hello. Here the skeletal muscle resulting the muscle prefers the fatty acids as a fuel, and during the high activity anaerobic respiration ensures. During the high activity a large amount of alanine is formed by the transmination of the pyruvate, and this is also sent to the liver as alanine, right? So it is a nitrogen. Then cardiac muscles, the heart metabolism is different from the skeletal. Heart muscle can function only under aerobic conditions. Heart muscle cells are rich in mitochondria, facilitating the aerobic respiration. This heart muscle is not able to store the glycogen. The cardiac muscle fatty acids are the preferred fuel of the heart, and glucose is the least flavor. The ketone bodies and the lactate are used under the stress when the energy demand. In relation to kidney, the major task of the kidney is the excretion. Water soluble metabolic waste products are created in the urine. The excess of water is removed under the osmolarity of the body fluids and is maintained. The final composition of urine is determined after the several cycles of the filtration and the reabsorption to avoid the loss of useful biomolecules. Yeah, the kidney generally energy required by the kidneys for this function is supplied by glucose and the fatty acids. The kidneys are a major site of gluconeogenesis, that is, liver is the major site. During starvation, the kidneys can contribute significant amounts of the blood glucose. The liver supplies metabolites and the fuel molecules to all peripheral organs. The liver cannot be efficient to use glucose or the ketone bodies, and the alpha ketone source as the energy activities. After absorption, by the intestine, the dietary fuels first destination, the liver. Liver is able to gauge the fuel availability and adjust the metabolic to the regulate the level of the various metabolites. Here you are observing the liver and the muscles are there. So glucose G phosphatase glucose develop to muscles, then glycogen gives to the glucose, alanine gives to the pyruvate and lactate. Glucogenesis is there. So pyruvate develop lactate and alanine, and the protein degradation is there. Here the amino acid metabolism in liver. The liver absorbs most of the dietary amino acid from the blood, and the amino acid are preferentially diverted to the protein synthesis rather than the catabolism. This is the possible because the kilometer for the amino acetyl tRNA synthase is lower than that of the catabolic enzymes. AE catabolism takes place when the anabolic pathway are the saturated. The amino acid metabolism in liver, it shows the capable of the sequestering nitrogen as urea for the disposal, and the alpha keto acids derived from the ether are the gluconeogenesis pathway. They serve as primary source of the energy for the liver. The carbohydrates and lipids metabolism, they shows the fed state, that is glucose, glycogen and fatty acid. Glucose used by nervous tissues, then RBCs and adipose and muscle fibers. The position of the century in the two earlier stages has glycolysis and the gluconeogenesis. In the carbohydrate and lipid metabolism, as we are knowing, they shows the such type of the characters are there. So here the one part is there, the obesity. Obesity mainly, as we are knowing, in most of the cases, obesity direct result of the overeating and the sedimentary. A sedimentary part is there. So here leptin is a protein synthesis by the adipose tissue, and it is called as the safety factor. The leptin's target organ is the hypothalamus, and this is the part of the brain which controls the food intake. The transport of oxygen in the blood. So we are knowing hemoglobin J402. So here we are knowing the red blood cells. Carbon dioxide and CO2 is important. So carboxyl is anhydrase, H2CO3, and later on it shows the diffusion oxygen in the plasma and CO2 in the plasma. So it gets diffused to the blood cell, and SCO3 generally in the plasma, and this one. So fat soluble vitamins, it shows they stored in liver and the adipose tissue. The body can't remove until they are used, and the toxicity takes years to appear but very harmful. It is important for the growth and the development, and they are ADEK, right? Then water soluble vitamins are there. It is not harmful with the excess dose, and there is having the will be removed by the urine, and they are composed of beta vitamins and vitamin C. The important for the metabolic pathways are there. The thiamine, precursor for thiamine pyrophosphate. Then fulvin, it is a precursor for FAD, and niacin, precursor for NAD. Plantothionate, it is precursor for the CoA biotic, and second of this, the coenzymes for the processing of the macronutrient. So it is all about the metabolic pathway of proteins in the therapeutic action, right? So you remember it properly. So we will see the next part in the next lecture. So thanks to all.